Hey everyone, and welcome back. Today we're going to talk about epigenetics. Epigenetics is a very interesting topic. There is a lot to it, but in this video, I'm going to try to give you a brief introduction to what we're talking about when we talk about epigenetics, how that impacts inheritance, and how that alters the expression of certain genes. So first, let's start by talking about what epigenetics really is. So epigenetics is the regulation of gene expression independent of the DNA sequence. So when we think about traditional Mendelian genetics, we think, okay, you get two copies of each chromosome, one from mom, one from dad. There's a weird exception when it comes to the sex chromosomes, X and Y. Sometimes you get one of each if you're a genetic male, or you get two copies of the X chromosome there. What's interesting is that simply having two copies of each gene doesn't necessarily mean that either of those copies will be expressed at all, or that one of them will be expressed and the other one won't. What I mean by that is there are cases where you may have two copies of a given gene, but you're only expressing one of them at any given time in a specific subset of cells. And this usually comes down to some type of epigenetic regulation of the genes that are involved. So when we talk about epigenetics, we are talking about the modification of chromatin structure. Now, hopefully you recall from some of my other videos, when we talk about eukaryotic chromosomes, they're not strictly DNA. They are DNA wrapped around clusters of proteins that we call histones. And we can impact the structure of chromatin by chemically modifying either the DNA and or the histone proteins themselves by adding certain post-translational modifications. The two biggest ones we're going to focus on in this video are methylation and acetylation. So we'll talk about how those can impact the expression of genes simply by restricting access to those genes by tightening or loosening the grip that the DNA has on those histone proteins. What's neat here is that this type of regulation can influence the expression of inherited traits without changing what's in the genes themselves. And we'll learn that today that this may play a role in cancer and possibly aging, and is also important for some types of inherited diseases. But it's also essential so that specific cells can express specific subsets of genes. Okay, we call that cell type specific gene expression. And this is really important in multicellular organisms. So I'll remind you that you, as a human being, consist of a few trillion cells, but each of those cells has their own identity. Some of that, when they belong to a certain tissue, they're going to have essentially the same identity as the rest of the cells within that tissue. But our liver cells need to express different genes than our brain cells, and our brain cells need to express different genes than our skin cells, which need to express different genes than our pancreatic cells, right? So how do cells choose which genes they're going to express and which ones they aren't? And at some level, that does come down to chromatin remodeling. This process is going to involve three broad groups of proteins: writers, erasers, and readers, and they are going to help to regulate and interpret what we now refer to as the epigenome. Okay, and the epigenome is the specific patterns of epigenetic modifications that are present in a cell at a given time. And this is what's going to produce the different epigenetic states that we're talking about. So here in this image, you can see what we're talking about when we talk about chromatin at the very microscopic level, past microscopic level. At the minutest level, you can see the DNA is wrapped around these clusters of histone proteins, and then these can stack on top of each other. And this gives us that structure of the chromosome that we see, like when we look at pictures of cells undergoing mitosis or meiosis. You see those neatly, tightly packed chromosomes, and that's what they look like when they're fully wrapped up in heterochromatin. We'll talk about what that means in just a few minutes. So first off, let's talk about the methylation of DNA. Okay, so this is often referred to as the methylome, which are the cells in the cell and tissue specific pattern of methylated nucleotides. Now, this is not a constant thing for the most part. This is going to change over time. Okay. And when we see this methylome kind of come into place and change, it's usually going to be after replication and when the cell is differentiating, when it's becoming the specific type of cell that it's meant to be. Now, in mammals, when we talk about methylation, we are almost always talking about the methylation of the 5' carbon of cytosines. Okay, this leads to something called 5-methylcytosine, which is abbreviated 5-MC. And this is almost always going to occur when that cytosine is directly next to a guanine in the DNA sequence. So a C directly adjacent to a G. You'll see these often abbreviated as CPGs. And this is going to occur through the activity of a group of enzymes called DNA methyltransferases, which for obvious reasons we often abbreviate as DNMTs. And what we actually observe in a lot of genes are these CPG islands. So these CPG islands are clusters of CG, 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 CG that exist in the promoter region directly upstream of genes. And what we can tell from looking at the methylome of cells and understanding gene expression a bit, what we find is these CPG islands are typically unmethylated in the promoter region of genes that are being transcriptionally expressed. So they're transcriptionally active. So demethylation is going to put that into an open chromatin state that's going to allow transcription to occur. However, methylation of upstream regions can then cause them to be transcriptionally inactive. So if we over-methylate these CPG islands, that's going to shut those genes down. So you can see in this little diagram, they've got these little like sticks with balls on top, and the darkly colored balls, those are going to be your methylated versions, whereas over here we have our unmethylated CPG island. This little green arrow, when we make diagrams about transcription, this means transcription is occurring. If we slash that off and there's like a vertical line at the end, that means transcription is inhibited at that point. So demethylation is going to encourage transcription to occur. What's interesting is when during reproduction, okay, when we see sperm and egg cells, typically the DNA inside of sperm and egg cells are highly, highly methylated. We don't want a lot of gene expression happening at that point. But what usually happens during embryogenesis, usually around the 200 cell stage, all of the methylation gets wiped out. Like basically, it's like an Etch-a-Sketch where they just shake it. I don't know if people know what those are anymore, but you shake it and everything's gone. And that's what typically happens with the methylome, is it gets wiped away because cells need to be able to differentiate and decide what their methylome is supposed to look like so that cell can take on its own unique identity. Typically, these CPGs are going to occur in the major groove. So if you recall from my video, from one of my previous videos on DNA structure, DNA has a major groove and a minor groove. That major groove gives you sequence-specific information. This is typically where transcription factors are going to bind to help regulate gene expression. Well, the methylation in these major grooves kind of sends the signal to the transcription factor of, don't bind here. This is inaccessible to you. We also see it in highly repetitive regions of chromosomes. So you may also recall me talking about the fact that only about 2% of our genome actually encodes genes that will ever become functional RNA products, but like 10% of our genome consists of lines, 10% consists of signs. These are almost entirely wrapped up in heterochromatin, which means they are always constitutively methylated. They are, please do not, there are no genes here, leave it alone, regions of the chromosome. You'll also see it at the centromere. So if you recall what the centromere is, it's that little waist region where the chromosomes are sort of joined together like that. So that's a region where we typically don't find genes to be expressed either. So they must be methylated as well. So methylation is great because it promotes chromosome stability. It can prevent, because when chromosomes are tightly clamped down on heterochromatin, it's hard for the DNA to intermix so you don't get translocations, and it can also prevent other chromosomal abnormalities. But it's also important that this process of methylation and demethylation is coupled appropriately. You don't want to methylate when you don't have the ability to unmethylate, and vice versa. And what we see when we look at various cells are cell-specific gene promoters are unmethylated in some cells where they're active, but the same genes may be locked down by methylation in other cells where they're not meant to be expressed. Okay, so for example, you can see that when we look at a subset, this is just a subset of genes they looked at, and you can see the number of genes that are methylated, and you can see there's differences. Of these 25 genes they looked at, the overwhelming majority are completely silenced in stomach cells, but the vast majority are active in other remaining tissues. And that's important for making sure that each of these cell types takes on their own respective identities. Now, the counter to methylation is acetylation. All right. So, and in some cases, phosphorylation. So here's what we'll talk about. We just talked about methylating DNA, but the histone proteins can also be acetylated, methylated, and phosphorylated. Okay. So for the most part, all of these modifications are going to happen on 3 different types of amino acids. It's either going to be a lysine, most commonly a lysine, whose amino acid abbreviation is K, or arginine, and in some cases, serine residues. And the reason why is they have the functional groups necessary for methylation, acetylation, and phosphorylation to occur. So when we talk about methylating histones, it's usually not the histone core proteins that are going to end up being methylated, acetylated, whatever. It's these tails. So when you look at these histone proteins, we often draw them in sort of these nice little ball shapes like you see here, but you see the tails sticking out. This is where all the action's happening when it comes to epigenetics, because these tails have lots of arginine and lysine residues that can undergo these post-translational modifications. And what actually happens is when these are in the closed chromatin state, these histone tails will clamp down and wrap around the DNA, or when they're in the open state, they'll loosen up. And that's really what controls how tightly wound the DNA is around these histones. Are these tails clamped down, or are they loose and limp and allowing the DNA to kind of float off of those proteins a little bit? Again, when we talk about the proteins involved, we're going to talk about writers, readers, and erasers. So our writers are often protein methyltransferases or histone acetyltransferases. Our erasers are going to do the opposite. So they'll be histone deacetylases, lysine demethylases, for example. And then the readers are these proteins that are able to read certain sequences, Tudor domains, MBT domains, and chromodomain proteins. That sort of their job is to basically tell the cell what state they're in. Oh, they're acetylated. Oh, they're methylated, and then kind of help that, kind of convey that molecule, or that help convey what the molecule's condition is. And one of the things I'll show here is when we look at a lysine, you'll see that nice amine group at the end. That is why we can attach an acetyl group to that, for example. All right. And you can see here what they're showing, just give you an idea of how the numbering happens. Actually, we'll talk about that on the next slide. So the way the numbering happens, there's a shorthand here. So first off, what's the histone protein we're talking about? Okay, it's protein H3. What type of amino acid are we discussing? It's K, so it's a lysine. What's the position? So it's K9. So K9 right here on the H3 histone tail. So H3K9 is the 9th amino acid on that tail. It's a lysine. What's the modification? Is it being acetylated, methylated, phosphorylated? You can see right here. And then there's usually a 3. How many? Okay, so for example, if we see H3K9ME3, that means that the lysine that's in the 9th position of the histone tail on histone H3 has 3 methyls attached to it. Okay, I know that's very confusing, but you kind of learn to read it once you're used to reading these things. So the people who are studying this, they know what they're talking about. You just kind of, you got to kind of go with it. There are other modifications you'll see down here too that I'm just kind of ignoring for the purposes of this. Like I said, there's a lot here. I'm just trying to do an introduction on how all this works. So when we talk about the histone code, this is important because we need to understand what's happening at each of these residues on each of these different histone proteins. And what's really interesting here is, when we talk about methylation and acetylation, we like to think in sort of oversimplification that, in general, acetylation it can lead to activation while methylation leads to repression. That's not exclusively the case, but we can sort of lump these things together so we understand what's happening. So, for example, acetylation is almost always going to lead to the activation. So you can see if you look at histone 3, if you acetylate lysine 9, lysine 14, lysine 18, lysine 56, that helps lead to the activation of that particular region of the DNA. You can see the same for these different histones. On the other hand, methylation often leads to repression. So if we're looking at trimethylation of K9 on the H3 histone tail, or trimethylation of lysine 27 on the same tail, or trimethylation of lysine 20 on H4, these are all repressive in nature. But you can see that in some cases, methylation can potentially lead to activation, but it's got to be under the right circumstances. Phosphorylation also seems to be important, at least in some cases. This is going to happen on a serine residue in this particular case. So it's important that we understand that while in general, acetylation is going to lead to acetylation of histone proteins is going to lead to activation, methylation can go either way. And again, I'm speaking specifically about the methylation of histone tails. Methylation of DNA, always going to be repressive in nature. It just is, okay? So we're just talking about how to read this language, understand what this means in terms of the context. And by the way, when we talk about activation repression, we're talking about the open chromatin state or the closed chromatin state. Open chromatin we typically refer to as euchromatin, whereas the closed chromatin we refer to as heterochromatin. Just a reminder, I know I've talked about that before, but I want to remind you since that's what we're really discussing today. So it also turns out that non-coding RNAs are heavily involved in this process. So this is why in genetic terms, we don't always like to say that a gene is a stretch of DNA that will eventually become a protein. In the overwhelming majority of cases, that is what's going to happen. But some genes never become a protein. They're never intended to. For example, genes that encode tRNAs, genes that encode rRNAs. Those are going to either be involved in decoding mRNAs in the case of tRNAs, or in the structure of the ribosome in the case of ribosomal, or of case of rRNAs, ribosomal RNAs. But there are lots of other non-coding RNAs that can be transcribed from DNA that don't become proteins. These are labeled different things. It's usually depending on their size. It can also be dependent on which proteins they interact with. So the names get a little bit wonky, but in general, if we talk about a short non-coding RNA, an SNC RNA, they are typically less than 31 nucleotides. There's always exceptions. Then we have long non-coding RNAs, LNC RNAs. Those are typically greater than 200 nucleotides in length. And these different non-coding RNAs are involved in heterochromatin formation, histone modification, site-specific DNA methylation, and in gene silencing. Now, we can break the short non-coding RNAs down into three different classes. We have our microRNAs, which are very short. Then we have siRNAs, which are short interfering RNAs. And then we have our piRNAs, which are named Pee-wee encoding RNAs after a protein called Pee-wee, which we found them interacting with. So, probably not the best way to arrange nomenclatures, but what we got. MicroRNAs and siRNAs are transcribed and form stem loop structures. This is what you're seeing here. Here's the stem, here's the loop, there's the stem again. And they get exported from the nucleus, and they get broken down into these little 20 to 25 ribonucleotide pieces, and they can inhibit translation, okay? On the other hand, these Pee-wee RNAs, they form an RNA protein complex, and they are involved in the epigenetic silencing of germ cells, all right? So different jobs for these different non-coding RNAs. When we look at the long non-coding RNAs, these look a lot like mRNAs, okay? They will have 5' caps and poly A tails. They end up getting spliced. So essentially, they get turned into a messenger RNA, but that messenger RNA never seeks to be translated by a ribosome, all right? They do not encode proteins. And what's really interesting is we have discovered, the number's probably higher now. This number is a little bit old. Last checked, there were at least 14,000 long non-coding RNAs in the human genome. So we may be well above that now. So this is one of those things where we find these and we're like, oh, wow. Yeah, that's a lot of long non-coding RNAs. They must be important for something. And typically, the way we classify them is based on their relationship to nearby protein encoding genes. Okay, so antisense. This will partially overlap the protein encoding gene, but in the opposite direction. So here's the gene. The gene is being encoded over here. This is the coding strand for this gene. I'm sorry, this is the template strand for this gene. But over here, where you would think this is the coding strand for this gene, nope, we're making a long non-coding RNA out of it. So the opposite strand, the strand opposite the gene, antisense, is actually a long non-coding RNA. We have our intronic long non-coding RNAs. These are inside of a gene's introns. They don't include the exons. They're just in those introns. So all that junk DNA, that stuff, like, why do we have all this? Why does it get cut out? There's one of your reasons for it. Sometimes we don't know the reasons, so we label things and say, oh, that's junk. Nope, not junk. Turns out it does a job. All right. Bidirectional, they will use the promoter of a protein encoding gene, but transcribed in the opposite direction. So here's the gene, here's the promoter. Well, RNA polymerase is going to land here, but instead of reading through and transcribing this gene, nope, it's going to read through and transcribe this long non-coding RNA going in the other direction. And then intergenic. These are located outside of any protein encoding genes. They have nothing to do with that at all. So what do these do? How do they impact epigenetic regulation? Well, some of them serve as guides, okay? So when you look at this, you've got these chromatin modifying enzymes that want to come in and do a job. They may want to add a methyl group, remove a methyl group, add a acetyl group, remove it, whatever it is. These long non-coding RNAs can help guide them to the right sequences. They go, oh, you're looking for where you're supposed to bind. Here, I'll find it for you. Then it lands on top of that long non-coding RNA and says, boop, this is where I'm supposed to do my work. So they literally act as pathfinders. That's their job, to go out and find these things. They can act as scaffolds. Sometimes they can sit there and allow other proteins to coalesce around them. We talked about how sometimes proteins form that quaternary structure where you have multiple subunits coming together. Well, sometimes that actually depends on there being one of these scaffolding long non-coding RNAs to be there. These sometimes are referred to as adapters as well. They can be involved in signaling or in the enhancer regions. They bind to DNA regions upstream or downstream, and they act to enhance gene expression. So they land and they help transcription factors land at the promoters or the enhancers. That leads to activation of the gene. So they can recruit some of these transcription factors so that the transcription factors can do their job. Or they can kind of do the opposite. They act as a decoy. So they will bind to a transcription factor that wants to land on a gene, and they essentially act like an enzyme inhibitor. They literally land here in the transcription factor, and the transcription factor is like, well, I was just going to go and promote the transcription of this gene. It's like, yeah, or not. We're just going to sit here because I'm basically making it so that you can't do your job right now. So these act as decoys. This will act to reduce gene expression. Okay. So what are the consequences? Like, why does the cell do all this? Okay. So why do we get this? Now, obviously, this is important for controlling which genes in our cells are expressed and which are repressed, allowing cells to settle on which sets of genes they're going to express and when. So there's a lot of this give and take just in normal life during the cell. But sometimes it can be a little more permanent than that. And there are three major ways where we end up with chromatin remodeling or epigenetics leading to something called monoallelic expression, or MAE. Okay. When we talk about monoallelic expression, what we are talking about is the fact that in every cell in your body, except gametes, set that aside, in every cell in your body, you have two copies of each chromosome. You have a paternal and a maternal copy of each chromosome. And in general, both of those genes, like if you have two versions of a gene, one from mom, one from dad, both of those are kind of like allowed to be expressed. And if you're homozygous for that trait, they produce identical proteins. If they're heterozygous for that particular trait, they produce slightly different versions of those proteins. But in some cases, that's not what's happening. In some cases, one of those chromosomes is just shut off and you're only expressing that one allele. Maybe you're only expressing the paternal version of that gene, whereas the maternal version is present, but wrapped up in heterochromatin and useless. So there are three major classes of monoallelic expression that we see. The first is the result of genomic imprinting. The second occurs randomly through these randomly generated patterns of autosomal genes. This may be as high as 10 to 20% of the genes in your body. And then we have the random inactivation of X chromosomes in genetic females to compensate for the dosage imbalance of having one sex have XX chromosomes and one sex having the XY chromosome. So we're going to talk about all of these in more detail right now. So let's start with imprinting. So imprinting is a process where genes are expressed in what we call a parent of origin fashion. That basically means, that in about 1% of your expressed genome, about 1% of those, either your mom or your dad's version of a gene is just automatically shut off based on where it came from. Just that's mom's. We don't express that gene from mom. So for example, when we look at the IGF2 gene, in general, the paternal version of that is what gets expressed, while the maternal copy of that is shut down through epigenetics. Just downstream of that is another gene called H19, and the normal imprinting pattern is for the paternal chromosome, IGF2 on, H19 off, which means even though you have two copies of your IGF2, you're only using your dad's. On the other hand, you have two copies of H19, but you're only using your mom's. However, there are cases such as Beckwith-Wiedemann syndrome, where this imprinting goes awry, and you end up with both IGF2s being expressed and neither H19 being expressed. And we see that with some other diseases as well, Angelman syndrome, Prader-Willi syndrome, as well as others. Okay, so what we see with imprinting is there is typically something called an imprinting control region near genes that will be imprinted. And this small 1% of the genome that is regulated this way is clustered together downstream of one of these imprinting control regions. And this is what gets methylated. Now, what happens, like I said before, the DNA of sperm and eggs are heavily methylated. Not a lot of gene expression happening. Their job is to find the other one, fuse together, and become the next human or the next whatever it's supposed to be. But as soon as shortly after fertilization, DNA gets demethylated. Like I said, we kind of wipe the slate clean around that 200 cell stage, and then the epigenome gets reset. Okay, right around this 200 cell stage, we get the wiping out, and then we get the resetting of it. And some regions escape this reset, and they retain that original methylation pattern. And these are the imprinted genes. Okay, so that's why, for example, in sperm, that IGF2 is unmethylated, but the H19 is heavily methylated. That doesn't do the reset. So because that chromosome came from the sperm and not the egg, that imprinting mark stays. And in mom, in the egg, that IGF2 gene is all heterochromatin, shut down. And that H19 is not, it's open for business. And that's where the imprinting comes from, okay? Interestingly, it does appear in some cases that both chromosomes can be imprinted, and that's where you get that kind of stuff happening, okay? Another example of monoallelic expression comes in the case of the female X chromosomes. So in human beings, as well as many other species, the differentiation between sexes comes when the female gets two X chromosomes and the male gets an X and a Y. There are lots of different ways in which sex can be determined in various species. But we'll talk about this one because, well, this is what we use. So let's just stick with what we have, right? So the problem you get here is males are hemizygous for anything on the X chromosome. They have one copy of each gene. So they can only express one gene and the product thereof. If the other sex is running around having two X chromosomes, there's going to be a dosage problem because they're going to be producing twice as much of all of those proteins, which can be quite problematic. So what was evolution's solution to this problem? Well, the evolution's solution to this was to basically, at some point during the developmental stages of a female, to just go, that one gets turned off in that cell, and that one gets turned off in that cell. How did you decide? No, I did it randomly. So in approximately if you are a genetic female with two X chromosomes, approximately half of your cells have your maternal X chromosome just completely wrapped up in what we call a bar body. And in the other half of your cells, it's your paternal chromosome that's wrapped up in your bar bodies. But this decision happens very early on in development. And what's really interesting is once that decision gets made, it's permanent. So once this cell has decided that the paternal X is going to get wrapped up and the maternal is going to get expressed, every cell that results from that, because mitosis, is going to have the identical epigenetic pattern with the paternal X completely wrapped in heterochromatin in a bar body. In the other half of cells, the paternal gets expressed, the maternal is a bar body, and then all of their descending cells through mitosis will have the exact same pattern. This is actually done through long non-coding RNAs. One of them is called exist, which is an X inactive specific transcript. That's what leads to the bar body. And because scientists sometimes like to have fun, the one that represses the expression of exist, leaving the X active, is known as T6, which is the reverse of exist. So, hey, we get to have fun once in a while. So that's how evolution has solved this sort of dosage problem so that the roughly 900 genes on the X chromosome don't get overexpressed in genetic females. This can also happen in autosomal genes. So we think this happens in as many as 10 to 20% of all genes. This is not clustering. So they're not clustered together. This is not imprinting. But basically what ends up happening is in a tissue-specific pattern, either both alleles or the paternal or the maternal or none of the genes, you get this tissue-specific spectrum of heterochromatin forming for these genes. And as a result, downstream of this, all of these resulting cells are going to have that same pattern of sometimes it's mom's gene being expressed, sometimes it's dad, sometimes it's neither, and sometimes it's both. So that's what's happening in each of your cells, which is really, really wild because as far as we can tell, this is entirely random. Now, I'm half confident that in 20 years, I'll be remaking this and we'll no longer be calling this random MAE. We'll understand why and how it happens. We're just not there yet. But that being said, if that's something that interests you, go research it. There are labs doing this kind of research all around the world. Find a lab, go study this. You got to find something to be excited about if you want to be a scientist and do research. So what are some consequences of epigenetics? What happens when they go wrong? So one of the things that we've been able to observe is that cancer cells generally have much lower levels of methylated DNA. What that means is that transcription is kind of wide open. There's no regulation there. And that seems to lead to unrestricted transcription of many gene sets. And one of the things we note is in these cancer cells, these sets of genes that seem to be left to kind of do whatever they want, to be transcribed whenever they please, these tend to be involved in cancer. So that seems to be a problem. The other thing we see in cancer cells, imprinting is gone. So where we had these imprinted genes that are on all humans, cancer cells go, we're done with that. We're demethylating these and we're just going to see what happens here. This seems to promote chromosomal rearrangements and altered chromosome numbers. That's why, again, cancer cells, all kinds of wonky, all kinds of bad stuff is happening. And what's interesting is there's also these regions that are hypermethylated. And surprise, surprise, in many cancer cells, that those hypermethylated regions, the ones that are tightly clamped down, cannot be transcribed, they're in the promoter of tumor suppressor genes. Like literally these cancer cells are using epigenetics to turn the brakes off. They are all gas, no brake. And that's why cancer is so problematic. This also challenges our older model of mutation-only based cancer. Typically, people think that you have to have at least we used to say 3. I've heard some people say as low as 2, but usually you need 2 or 3 different mutations to turn a cell into a cancer cell. Okay. And one of the things we're trying to do is, can we get new anti-cancer medications that reverse these epigenetic changes? We've also seen that aging cells begin to show hypomethylation. All right. Lower methylation. And one of the things that is kind of an open question in the field of aging, is this hypomethylation a cause of aging, or is it a scenario where this is a symptom of an aging cell, right? But one of the things we think happens is that when you have all of these regions open for transcription, it probably makes it harder for the cell to focus on what it's supposed to do, right? There, instead of being saying like, I can only transcribe these genes because these are the only ones available, we've kind of lost that regulation and we get a lot of transcriptional noise showing up. So, this could be problematic for a lot of different reasons. So one of the things we are now learning is that it's possible that epigenetic traits are heritable. So recent studies have shown that epigenetic changes can actually be inherited from not just from mother to child, but even down to the level of the grandchild. Okay. One of the observations that we've seen is that children of women who are pregnant with that child during times of famine, we have observed higher levels of schizophrenia and other psychiatric disorders in these children, higher rates of obesity, incidence of heart disease and breast cancer also seem to be high. And we've actually seen that some of this has persisted into the next generation. The study that looked at this was done on, I believe they were Dutch women who were pregnant during the Nazi occupation in the 1940s. Many people don't know there was a huge period of famine during that time because basically all the food was being used to feed the German people and the German soldiers, and the Dutch just kind of had to fend for themselves. But the subsequent generations that were born from these women have these kind of the higher rates of these traits. And this corresponds with other research that's shown that epigenetic changes happen in people suffering from Alzheimer's, Parkinson's, Huntington, and schizophrenia. So it's possible that these inherited epigenetic traits are leading to higher incidence of these conditions, diseases, whatever you want to call them, in subsequent generations. And this may help to explain the variation in terms of the onset and the severity of the diseases. So how they happen, how severe they are, this may contribute to that. Interestingly, they've started to do some actual scientific studies on these. One particular study looked at rat mothers and their pups, and they broke them down into what they would call an anxious, low nurturing mother or a relaxed, high nurturing mother. And what they found is that the pups of anxious, low nurturing mothers are much more likely to become an anxious adult, whereas the pups of high nurturing mothers are much more likely to become a relaxed adult. And one hypothesis that came out of this study was that it may be the result of low glucocorticoid receptor expression due to hypermethylation of the promoters of the genes that encode these proteins. So I'm not going to get into the full depth and breadth of this study, but kind of cool that we're actually being able to observe this in a laboratory setting. And we're starting to begin to understand that epigenetics isn't just something that happens in you. It's actually something that can be inherited like any other gene allele or something like that. So very cool information there. So that is what I have for you today as sort of an intro to epigenetics and how it works. I hope you enjoyed the video, and I hope to see you again real soon. Thanks.